college. So I'm Professor Pantelis Georgiou. I'm a IEEE Census Council Distinguished Lecturer, and my talk today is going to be focused on what you can see in the title. So using microchip technology to create a next generation of rapid diagnostics. And I've gotten the title from AMR to COVID-19 because I've also been telling you about my journey in diagnostics, which started in the realm of addressing antimicrobial resistance and later on during the pandemic for COVID-19. I am sponsored by the Census Council, so I need to give just a brief background on the Census Council. So the Census Council is an IEEE entity devoted to sensor theory, design, fabrication, manufacturing, reliability, and applications of devices for sensing and transducing physical, chemical, and biological phenomena with emphasis on electronics, physics, and reliability aspects of sensors, integrators, actuators. It is not a technical society as in the conventional sense. It's made up of many technical societies within the IEEE. That's why it's a council. So it's made up of many IEEE societies. However, members can register as participants and does a variety of activities such as conferences, publications, technical activities, standards, education, and networking. At the moment, it's made up of 26 member societies of the IEEE, and it's in many global locations. I mean, it has an annual conference which rotates every year, and 35 section chapters with 29 student branches. Our flagship conference is Sensors, which has happened in Vienna. We also have FLEPS, Inertial, SIM, ABSCON, and more recently we launched IEEE Biosensors Conference. I launched it in London in 2023 with Mike McShane, and our next meeting will be in Cambridge this summer. It also has a journal, the Sensors Journal, letters, and many co-sponsored publications, a YouTube channel with a variety of education, and here you can find more details on the website and some of the social media aspects in relation to the council. Now back to my talk. So this is the world we currently live in. I mean, we've just faced a massive challenge, the global pandemic of what was COVID-19. And it changed everything. I mean, we learned a lot in terms of what the potential for diagnostics could do. And we all had our independent strategies in trying to deal with it, but we also saw that the role that diagnostics played in addressing COVID-19. I mean, here in the UK, we had what was called Operation Moonshot, where the target was about 10 million tests per day. And the opportunity was clear. If we could diagnose COVID, if someone was positive, then we could isolate, not infect our loved ones and other vulnerable groups. But also, if it's more importantly, if it's not COVID, then we know it's something else less, well, hopefully less severe. And then we go about finding the best way to manage that infection. But we all, at some point, I think in the last 3 or 4 years, woke up similar to this person on my screen. We woke up with a bit of a runny nose, a bit of a fever, and we said, "Oh my God, is it COVID?" It's a simple question, but if we had a device like this device, which was very popular around the time when I was a teenager, simple handheld device with a small cartridge, you could plug it in and it could answer that question. More importantly, it could not just say whether or not it's COVID, it could tell you if it's not COVID and whether or not the infection, for example, is a bacteria or a virus. It's a simple question, which has massive implications because it can then tell you, do I need to take antibiotics or not? Obviously, antibiotics works on bacterial infections. If it's viral infection, you don't need antibiotics. And therefore, you don't unnecessarily take antibiotics, which will then contribute to this global problem of antimicrobial resistance. But I'm hoping to convince you in the next 40 minutes or so, the device like the one I'm showing my screen is no longer science fiction. It's more like a reality. And we can achieve that reality because of the advances that we're seeing in semiconductor and microchip technology. So microchip technology, as you can see on my screen, it's revolutionized consumer electronics and computing. Fundamentally, we make a semiconductor silicon microchip that you can see on the left here. Let me just put my laser pointer. That you can see on the left here. And that you can see on the left here. And fundamentally, it's made up of millions, if not billions, of these transistors. So transistors, I say microchips, but let's say nanotechnology, that refers to these transistors, which now are at the nanoscale. We're down to about 3 nanometers in the latest Intel microprocessors. I mean, there's more transistors on a semiconductor chip now than there are humans on this planet. But in my lab here at Imperial College, we use the semiconductor chip as a platform technology to innovate new types of medical device and healthcare technologies, because with it, we can integrate now chemical sensing, instrumentation, and communication to make fully integrated system on chips, which can go anywhere. And fundamentally, my labs focus on 3 pillars. The first is bio-inspired design, where we look to create systems which replicate biology to treat chronic disease. And here you can see an artificial pancreas, which we made for management of diabetes. On the bottom left is the integrated sensing systems, where we look to integrate thousands of electrochemical sensors on a simple chip to make what's called a lab-on-a-chip device with microfluidics. And then we use that as a diagnostic technology. My talk today is going to focus on this arm of my research. Combining the two, we also do a lot of wearable devices where you look to put stuff on the body, which monitors in real time more power efficiently, and it runs edge machine learning and communicates to a mobile phone to give you meaningful data for management of disease. The other side of my research, however, is focused on addressing unmet needs where technology can have the most impact. So a lot of the research I do is in collaboration with low middle income countries, because there they don't have access to sophisticated healthcare technologies. I mean, take, for example, outbreaks in malaria and dengue, which happen annually. I mean, we experienced a pandemic in the UK, and in the countries in sub-Saharan Africa and Asia, they experience these types of pandemics every year. And because people are very dispersed, it could take days, if not weeks, to get access to healthcare facilities to, for example, provide a routine diagnostic test that could rule out a certain type of infection and then inform the best type of treatments. So I've been working hard with partners in sub-Saharan Africa and Asia to bring diagnostics to the front line to address these unmet needs. And as an engineer, I like to design to the specification that's given to me. And thankfully for diagnostics, the World Health Organization has given this specification on what a diagnostic technology should conform to. It's called the ASSURD criteria. It should be affordable. I say they're less than £10 per test so that it's readily deployable. Sensitive and specific so that it can detect exactly what you're looking for with very, very high specificity, meaning it can detect very low levels of infection. It has to be user-friendly, and I use this as a message to my lab as well. We can design the world's best technology, but if nobody wants to use it, it's not going to have any impact. It needs to be rapid, obviously, because you need to, knowing that the result knowing the result quick can have huge implications in terms of treatment and spread. It needs to be able to sit on a shelf for at least 2 years, let's say, because you can manufacture diagnostics, but then if you've got a short lifetime, then it's not going to have any usability for deployment. It needs to work without any sophisticated equipment and deliverable to end users. And if we look at the current diagnostic techniques and what's currently in play within diagnostic facilities, the conventional method is which is like a gold standard. You can see on the left where you culture things on an agar plate. You need a dedicated lab where you send off a sample and you test for susceptibility. Need specific expertise, and it can take up to days, if not weeks, to get a result. However, it is definitive. You are sure of what you find, what you're looking for, its sensitivity, and it is quantitative. On the other side, during the pandemic, we became very familiarized with these types of tests, the lateral flow rapid diagnostic tests. And they were very good because they could give us an answer in under half an hour, very cheap, very deployable and portable. However, they weren't very qualitative. I mean, they couldn't tell us how much infection we had. And also, they were prone to a lot of false negatives because the window of detection in terms of the viremia was very narrow. We became familiar with this during COVID, that by far the best technology was the confirmatory PCR, polymerase chain reaction technology, because it had very high sensitivity and specificity, and it was quantitative. However, you did need to send a sample off to a lab. The instrument would take a couple of hours to diagnose what the infection was. Needs dedicated expertise. The instrument that you can see in the picture there, which is standard PCR, costs about £50,000. So it's not really deployable at the point of need or the point of care. So my premise is that the best diagnostic technology combines these two worlds. So it's got the sophistication and advancement that you would expect of a PCR technology in a lab, but it's cheap, portable, and deployable, similar to the lateral flow. And we can realize that with microchip technology. So microchips, I say here, are suitable for DNA diagnostics. Why? Because with microchips, we've shown that we can do label-free detection without the need for any optics of nucleic acids, DNA basically. Therefore, you remove the need for any bulky optics that would otherwise need in a PCR machine, so you can make something very, very small in terms of handheld. It gives us real-time detection capability. And what does that mean? Underneath our electrochemical sensors, we can also put our instrumentation. So it's a full system on chip. With that comes the opportunity to make things that can detect things at very, very low noise levels. That really boosts our limits of detection, which only improves our diagnostic performance. Thirdly, we can integrate thousands of sensors on a single substrate. That's important later on as I show you some of the advances that we're making in terms of multiplexing. So with a single test cartridge, you can look for a variety of infections. And we've proven that we can detect DNA with microchips using transistors, similar to the ones I showed in my first slide, that nanotechnology, but we call them ion-sensitive field effect transistors, okay? So what are ion-sensitive field effect transistors? They're standard transistors, as you would normally expect, with a gate, a source, and a drain. And we extend the gate to the top of the chip. And then with an insulating layer, we make it sensitive to ions, hydrogen ions. So it's pH sensitive. So effectively here, we've made a chemical transistor whose voltage, specifically its threshold voltage, is proportional to pH. So it's a pH sensitive transistor made using conventional semiconductor technology in CMOS. So effectively, all the circuit design techniques that we've been innovating for the past 50 years can be applicable to this chemical transistor. And here you can see a first example of a chip we made with 5 ISFETs that were quite large in this instance. But you can see what the performance looks like. So this is your standard ID VGS curve that you would expect of a transistor. And as we change the pH, it shifts. So what's important then is to design instrumentation to track that shift in pH, and then you can check what the pH of your solution on top of the ISFET is. But you may ask, pH is not DNA, so how do you detect DNA? Well, the key there is DNA is double helical, so it binds always to its complementary strand. So if you're looking for a specific target DNA, you'll design what's called a primer that's complementary to the target that you're looking for. And that's how you identify the presence or the absence of a specific DNA molecule on top of the chip. Then in the presence of what are called nucleotides, the NTPs, and an enzyme called polymerase, a chain extension happens on this DNA. And every time there's a chain extension, a byproduct of that chain extension is the production of a proton. So this reaction releases lots of protons. And the net outcome is, if my sample has my target DNA, there's a pH change, which changes the global pH of the solution. If not, there isn't. So we made a pH-based detection mechanism for nucleic acids, which we're using specifically for DNA and RNA. And what's the advantages? The advantages is that you don't need to label any of your targets. So conventionally, you put some labels, shine light, and use a camera to see fluorescence. We don't need to do any of that. It can be freely floating in the solution. With it, we can incredibly simplify then the sample preparation process. So it's label-free. We can implement it using thousands, if not millions of sensors in CMOS. So it's an incredibly scalable technique. And there are commercial platforms available now. On the left, you can see one we spun out from here in Imperial College, which looked at single nucleotide polymorphisms, genetic mutations. And on the right here, this is the Ion Torrent full genome sequencing machine, which is revolutionized next generation sequencing by simply having a chip with an inlet and an outlet, and then you flush a solution with a sample, and then you can detect what DNA is present in that sample. But in my lab at Imperial, I mean, we've been innovating this for the last, it's almost 20 years now. It's coming up on, I think, 18 years, where we fundamentally showed that you could do and produce ISFETs in unmodified CMOS. And we've evolved our platforms all the way to now getting really, really high scale arrays, which allows us to do many, many interesting things, which I'll talk about. But I'm showing this slide here to tell you, it's not just been a journey about creating microchips. We also have to consider the fact that we're doing something unconventional. We're putting a liquid on electronics. So there's an exercise here where we look at packaging and microfluidics. And also the ISFETs are potentiometric sensors. So we need to deposit metals to it to apply a silver chloride reference. But what we can do now, and you can see here, this was one of our earlier chips, is things like this. So whoever I show this animation on the right and ask them, what is this? They'll tell me, oh, is it the output of a chemical camera? And I say, no, that's the output of this chip. It's 1,000 electrochemical sensors imaging in real time a chemical reaction happening on the surface. So we're revolutionizing the way that we can look at electrochemistry, and we're calling it ion imaging. And we've broadened this out. Now you can see here an example made by Nick Moser and Chiara, my PhD students, where we can do multi-ion imaging now, where we can not just detect pH, we can put ionophores and look for a multitude of ions from a simple reaction. And then we can use things like machine learning, because it then becomes almost like an image classification problem, to decompose the mixtures of ions on these chips. Okay. And we're pushing the limits. So here you can see some work from my student Zhengming, where we're measuring electrochemistry faster than the human eye at 3,000 frames per second, where you can see clearly diffusion electrokinetics from a chip which is doing the ion imaging. And some of the recent applications that we've been able to show with this in collaboration with Martin Bertol in bioengineering is we're actually characterizing spatial temporally now anything that's on the chip electrochemically. So in this example, we could spatial temporally characterize ionic membranes and optimize them to give us specific measurement of potassium, sodium, and pH on the surface, because we're doing a project where we're looking at detecting very, very fast transients of potassium as they're happening in the body. So what do we feel like with this technology? Well, I almost feel like when Galileo pointed his microscope to the moon and started discovering, what the intricacies of the moon were and what the details were. We're at that stage now with these chips where we're really unlocking insights to electrochemistry, but we're also moving into more advanced domains. And here you can see a collaboration with the Institute of Cancer Research and Chris Pakal's group, led by my PhD student, Elena Beku, where we're now growing cancer cells on these chips and looking at the electrochemical profiles as we test the response to chemotherapeutic agents. So lots of interesting avenues and applications happening. So I've sidestepped from infectious diseases to show you what's going in the lab, but now coming back to infectious diseases. Detection of infectious diseases in terms of molecular detection, similar to PCR, is all about detection of the nucleic acids. And while I talk about DNA, it's the same concept because it's the same chemical makeup for RNAs like COVIDs. We can look at bacteria, gram-positive and gram-negative, fungi, protozoa, plasmodia, for example, like malaria as well. But one of the challenges of detection of infection is the sample preparation, because you have to detect in some instances the DNA which is inside the blood and inside the cells. So what you need is a chemical process called lysis to break open the cell. You then need a cleaning and purification, an elution step, and then you get your extracted sample, which you put on the chip. But by that time, the number of copies of DNA that you have might be incredibly low for you to be able to detect them. And that's effectively why we use PCR, polymerase chain reaction. It's a well-established technology. And the way PCR works is it thermocycles between two temperatures, 60 and 90. Why? Because at 90 degrees, it's the temperature at which DNA denatures, so it breaks open. So if you cycle DNA to 90 degrees, it'll break open. Then in the presence of nucleotide, it will extend the DNA single strand, and it will replicate. So every time we cycle between 60 and back down to 90, we get a doubling in the number of DNA. So that increases the concentration of DNA inside your sample, and it then makes your pathogen more detectable. And as an example of an integration, we showed in 2013 in this chip, which we published in Nature Methods, that we could do the full integration. So with the CMOS chip, we had the ISFET sensors. We put on heaters to do the thermocycling, and we put on temperature sensors to be able to detect what the temperature was. And we coined this term, we called it then pH PCR. And we showed its utility for looking at genetic mutations, single nucleotide polymorphisms. These are single base genetic mutations inside your DNA, which what the advancement of detecting those has become is it has a specific focus in personalized medicine, in that if you've got specific mutations, it can predict your response to drugs. And in this example, we were looking at warfarin. It's the blood thinning agent, because it's been known that if you've got this specific gene that I'm showing here, the specific mutation, if you take warfarin, it can cause you to bleed to death. So we're looking now at an area where we're using companion molecular diagnostics to be able to tailor the specific drugs that we're giving to patients. So that was 2013. The lab's moved on a lot since then, as I said, coming back to this vision I had of the handheld molecular diagnostic. And I'm putting this slide here to make you appreciate that it's the sum of many parts, all of which need to come together. It's not just about how you extract your sample. You need an extraction technology. You need a nucleic acid amplification step, and I'm going to talk a bit about how we've advanced that beyond PCR. You need your ISFET sensors. And I'm also a big advocate of the digital diagnostic technology. So because we have the capability to connect our diagnostics to the cloud, we can also do real-time surveillance of any infection. I mean, the COVID-19 surveillance was fantastic, as we saw these maps showing the outbreaks in different regions. That's something that we were doing way before the pandemic for things like malaria in rural areas like Ghana, and applicable now to current modern age diagnostics. So to facilitate that, we're using this chip. We call it Titanics. Primarily, it was made by two of my PhD students. You can say they're Nick Moser and Nick Misk. The array we're using in infectious disease is the one on the left, made by Nick Moser. And it's a very unique electrochemical sensor, because leveraging also CMOS technology, we're embedding inside each pixel processing capability. So it's a 4,000 electrochemical sensing array, and it can also compensate for any non-idealities in the sensor because it's adaptive. It works in a notion which we call pH to time. So rather than encoding the signal amplitude in a voltage or a current, we encode it in a digital pulse. In doing so, we've made a fully digital pixel, which is scalable to any CMOS technology. We've shown it working in 350 nanometer. We've also shown it working in 180 and 65, primarily because the amount of analog in this pixel is limited to the front end. Additionally, we've got a compensation capability to continually offset and compensate for any pixel non-idealities. And because in our chemical sensor, you can see it here, chemical sensors need to be relatively larger than the actual ISFET transistor, primarily dictated by the microfluidic requirement, because there's no point in making a sensor which is, for example, 3 by 3 nanometers, because you can't get that much volume on inside the channel. So we go for slightly bigger, but then with that, the opportunity lies in that we can also embed circuitry within our pixels. And in this example, we embedded a memory cell, an SRAM. So each of the pixels for the sensor also has embedded within it a digital code, which we use to compensate for any non-idealities. So it's a fully robust and dynamic pixel architecture. And then for the nucleic acid amplification, I talked about PCR, and we've pivoted to LAMP, loop-mediated amplification, because it allows us to amplify the DNA using a single temperature. We only need 63 degrees. And the way LAMP does that, unlike PCR, and this is through my collaborator, Jesus Rodriguez Manzano, in the Faculty of Medicine, is that we use basically 6 primers and a chemical enzyme that breaks open the DNA, and the DNA replicates itself by looping over itself. That's why it's called loop-mediated amplification. With the 6 primers, though, we get really, really high specificity, and it's also incredibly fast. So we can amplify the DNA in under 10 minutes. Also, more importantly, because it's isothermal, 63 degrees, it lends itself more readily so that we can make a fully portable platform. So combining the two then, we've got the molecular methods which we make to detect our targets, and we combine molecular biology with electronics. The electronics has to be a microchip with the microfluidics sitting on a gold substrate and bonded to a standard printed circuit board. And this is it. You can see a single-use cartridge here. It's also got the chip. We use standard consumer electronics here. So I say we've leveraged the £1 trillion investment in consumer electronics to make a very, very cheap and disposable cartridge, which we can use in low middle income countries. It contains the PCB, the chip, and a 3D printed manifold, which we can also tool and use using low-cost plastics now. So the actual bill of materials of this cartridge is actually relatively cheap. What's expensive is putting all the pieces together on a production line. But obviously, as you try and scale it, then the costs come significantly down. So here's some example of 3D printed microfluidics that we've done, pioneered by Matthew Cavuto, who's also a PhD in my lab doing mechanical engineering. And you can see here a two-well inlet system on the chip where we can put a sample and a control. Pulling it all together, we create Lacewing. So Lacewing is our handheld molecular diagnostic for infectious disease. It contains our single-use cartridge. You plug it into the handheld reader, and in under half an hour, it amplifies and it detects your DNA. And then in real time, it connects to a mobile phone. So we get real-time surveillance and mapping of any infection as it geotags the results. And how does it compare to the existing technologies? I mean, you can see here a like-for-like comparison. So we are definitely a lot cheaper than your standard PCR, where you would need about £60 per test in this £30,000 instrument. We're very, very rapid, so we can detect it under half an hour, where a PCR would take 2 hours. We do not need the lab. We can take it anywhere. It's quantitative, so it can tell you exactly how much infection you have in your sample. And why that's important, because if you take 2 time points of the infection, if it's going up, you know you're at the early stages. If it's going down, you know you're on the way out, so the treatment's working. And we can do real-time reporting. How it compares to lateral flow, it's never going to be as cheap as some of these paper-based lateral flow microfluidics devices. However, with opportunity that we can detect multiple pathogens, in our latest chip, we can detect 10, you could easily do the equivalent of 5 different lateral flow tests with a single test. And bearing in mind that we're equivalent to a molecular PCR test, there is added value to using our lab-on-a-chip cartridge. It's as quick, it's very easy, it's got a lot larger sensitivity and specificity, and it's quantitative. And it can do real-time reporting as well. So it's got its place in this world where we look to make something more advanced than lateral flow, but something fully portable, like a PCR. Okay, and here's a video example of the form factor. This was a prototype that we made for the pandemic, which I'll speak about our journey during the pandemic in a second. But you can see they're about the size of a mobile phone. You plug in the single-use cartridge. If you pair it to the mobile phone, you see the output of the 4,000 sensors. Give it the sample. And you give it the control. And then you see things changing in real time on the screen. So as I mentioned, one of the key challenges we were addressing in the UK was antimicrobial resistance. So because we're unnecessarily prescribing antibiotics, you know, any infection specialist would probably start with a broad spectrum antibiotic to treat something which he suspects could be bacterial, but it's also most probably viral. The consequence of that is we're driving up how resistant these bacterial infections are to antibiotic. As a consequence, antibiotics are ceasing to work. So they're predicting by 2050, this was the famous Jim O'Neill report here in the UK, that by 2050, there'll be more deaths from antimicrobial resistance than there will be by cancer, because we're facing a future where antibiotics will stop working. They're estimating about 10 million deaths per year. So we need strategies to be able to combat antimicrobial resistance, and the role that diagnostics can play. In the first instance, as I said, to rule out the unnecessary prescribing of antibiotics. In the second instance, if it is a bacterial infection, to know exactly what the resistance strain is, so then you can tailor or target this with a specific antibiotic that you know will work for that resistant strain. And here in this first example, we piloted it with an Enterobacteria's resistant strain. Like for like with this £50,000 Roche instrument, we showed that our handheld device could amplify the DNA. You can see good separation from the sample and the control. And we began to see clear separation just after 5 minutes. So that's why I say we can detect it here in 7 minutes. So that was our first demonstration for antimicrobial resistance. Moving forward, I talk about the chip being quantitative. And what that means is we can also tell you how many copies of DNA you have in your sample. And I explain why that's beneficial, because you can tell, for example, if the infection's clearing, you can get a nice time series of your infection dynamics and viremia or bacteremia during its time course. And that also informs treatment strategies. But here you can see we tried it for Klebsiella pneumoniae, and you can see a nice here, we call this a standard curve. For different copies, we could detect down to 10 copies per reaction with our LAMP assays. Obviously, you can appreciate the less number of copies you have, the longer this is time to positive, the longer it takes to amplify. So it's by no surprise that if you've got a lot of copies, it can detect it really quick in 5 minutes. Less copies, 10 minutes. But more importantly, it's still under 10 minutes, and we get a nice separation of the sample and the control. So we showed that we could detect bacteria reliably. And then we were challenged. I mean, we talk about the pandemic. So now I'm going to start talking about dates. I mean, pandemic, it happened early 2020. Back in November of 2019, within our hospitals here in the UK, specifically one of our biggest ones in the NHS in Hammersmith, through my collaborator, Alison Holmes, who's a leader in antimicrobial resistance, they began to detect the emergence of a new resistant strain to colistin, the MCR9 gene. And while they were detecting this new emergent strain, we were tasked on creating a diagnostic for it. So we rallied the lab, and within 3 weeks, we rolled out a diagnostic technology which could detect it. You could see there in under 10 minutes. And this was a huge exercise for us, because we showed also how versatile the technology was, that we could rapidly repurpose to any emerging pathogens in a very, very quick amount of time. So while this was being reported to Public Health England, we were sending the paper to Nature Scientific Reports, and it got published to show the capability of the device. So that was important for us moving into the pandemic, because we knew that once the pandemic happened, we should undertake a similar exercise of repurposing the diagnostic. Obviously, in November, we didn't know there was going to be a massive COVID-19 pandemic. We were doing other things as well. So with collaborators, you can see Jake Barn and Aubrey Cunnington, we were addressing this global burden of malaria, looking at detecting malaria species, because there's a huge unmet need of detection of malaria in sub-Saharan Africa, where there's about 400,000 deaths per year. And we were detecting malaria, but also looking at the resistance to anti-malarial drugs, such as artemisinin, which could be life-changing. And with my PhD students, Kenny Malpardidis and Nick Misgoudis, we showed the technology was versatile for malaria, and we could also do on-chip quantitation. Summer of 2019, the team were in Ghana. You can see here a nice, the video is not playing. So this is a team in Ghana, and what we're doing here is we're showing that we can detect malaria species in under 15 minutes. And this was also a first exercise for us, in that we showed that the technology could travel outside of the UK to an entirely different context, and we could diagnose infectious diseases. So Ivana, Panisi, and Kenny there doing our trial for malaria. That was in the summer of 2019. And the plan was, and this program is part of what we call the Digital Diagnostics for Africa. You can see our website on the bottom. The plan was we were going to go back in the summer of 2020. We were going to put the diagnostic in the back of a vehicle and drive to rural Ghana to detect asymptomatic malaria in children in schools, with a view to containing spread. Obviously, we all know what happened in the summer of 2020, so that mission got put on hold. But thankfully, now we've been funded by the NIHR to produce what is called the Global Health Research Hub, and the mission is back on track. I mean, we sent first devices for malaria just last month, and the program now is continuing post-pandemic. But we've been working on a variety of infectious diseases. I mean, last week I was in Thailand, where we were diagnosing dengue, for example, as well. So we've shown that it can work for dengue, malaria, antimicrobial resistance. We've got a program with South Africa looking at tuberculosis in children from a blood-based test using what's called host transcriptomics. Locally in the UK, we're also looking at a fungal infection called Aspergillus, which is deadly for for people that are admitted to hospital. And we're even using it in animals as well, looking at diagnosing infection in pig and livestock, contributing to this One Health approach for addressing antimicrobial resistance. So we were doing all these tests, and then COVID-19 happened. So I showed you what we did in the summer. I showed you what we were doing in kind of November of 2019. Then fast forward to January, we start to hear what's happening in Wuhan. Everybody's trying to figure out, should we or shouldn't we lock down? Then specifically in the UK, we locked down in it was the 15th of March, 2020. I remember it well. Government said, go and stay home. The university campus shut. I mean, as academics, we were also tasked with, pivoting to online teaching, so it was mad. For us, I mean, I remember it vividly because my son, he was just turning 1 year old, and we were set to go for Father's Day. In the UK, it's on the 18th of March, and we were set to go there for Father's Day, and they told us, no, stay home. So we all got locked at home. The nursery closed, so we had to do daycare as well with my wife, and it was just this crazy time. But at that time, I rallied the lab, and I told them, look, this is happening, but we've got something powerful here that can address COVID-19. So thankfully, we all became, in the UK, we called it key worker status. So we got a letter from our then president, Alice Gass, and we kept the lab going at Imperial. So if you would come to our South Kensington campus, it would all be pitch black, as you can see, and there'll be a little light open in the Bessemer building where our lab is. Where credit to the fantastic team, they kept working throughout the pandemic. And also, what was good for us during that time. So Imperial College, we've got 4 of the biggest hospitals in London. So we also became a centralized testing facility for all of London during the pandemic. So we also had a very rich sample source, which we had direct access to, which is some of the benefits of working in a multidisciplinary environment, like the one that we have in Imperial College, where we have readily access to our hospitals and our clinical expertise that can help us truly innovate in a multidisciplinary environment. So that was our task. We wanted to do SARS-CoV-2 in under 20 minutes using the technology. And we showed it. So using that same chip with the 4,000 sensors, we did a trial where we showed that we could detect it in under 20 minutes with a limit of detection of 10 copies per reaction. You can see here some of the outputs of the chip from a clinical sample and a control, and it gives you the positive outputs and the negative. And we ran it then on this was the first type of validation we had 183 samples. You can see here a nice linear correlation with the qPCR machine, and average time to result was 12.9 minutes. So we hit that target in under 20 minutes with a pretty decent sensitivity and specificity. I mean, back then it was 100 specificity and 90.5 sensitivity. But also, I mean, we've moved we've really advanced it since then, because you appreciate a lot's changed. So now we're more close to 100%. So we were really redefining molecular diagnostics. I mean, I show this figure here because I get asked a lot, how is it different to like the rapid diagnostic antigen tests? Well, simply here you can see, think of the blue curve as the amount of COVID you have when you're infected. So this is your exposure, and this is when your PCR would detect it. Okay, and then you develop antibodies. Your lateral flow, it's got a short viremia, so I say it detects it only here. So in this phase here, where you're asymptomatic, just before symptoms detect, your lateral flow be negative, but you can still spread the virus. Whereas our technology can even detect it here. Okay, so it's incredibly sensitive and specific technology, which can detect infectious disease similar to a PCR at any time in the infection cycle. And we also had our real-time surveillance system, building on the work we're doing in Africa, showing our trends and where things were happening in the UK. And that was our dream. And anybody that watches Scooby-Doo and then the Mystery Machine, we were going to run it in the lab in a boot, have it in the back of a car driving around, because it's a versatile, portable technology that can travel anywhere and give you molecular grade diagnostics, similar to what you would expect in a standard laboratory. So what have I shown? I've shown that you can make molecular diagnostics, which you can carry in the palm of your hand. It's a rapid molecular test which takes just 20 minutes. It's sensitive, specific, and quantitative. It's a fully mobile platform which can geotag your results and gives you real-time surveillance. It's lab-free, low cost, and it's incredibly scalable, okay? So you can adapt it to new pathogens within a month. And what happened next? Obviously, having proven the technology, Imperial College then got behind us because we got called upon not to make hundreds of devices, to make thousands of devices a day. So we spun out a company called Proton DX, and its vision was to, and is, to create rapid diagnostic technology that you can take anywhere. Obviously, the name Proton comes from the nature of our detection technology, which uses protons. So we're scaling up and manufacturing the device, creating a new next generation lace-wing device, but also continuing all the work that we were doing with our low middle income partners. So I'm pleased to show the latest prototype. You can see here, this is our lace-wing handheld reader. We now have a 60,000 electrochemical sensitive array. And we can detect in under 20 minutes, we've got a full panel on the chip now, because it can detect 10 different targets. So we can detect whether or not just if it's COVID, we can look for flu, rhinovirus, RSV, strep, and a variety of other infectious diseases. This is our diagnostic cartridge, which also has the chip. It's a single-use cartridge. Inside here, we've also got our proprietary extraction technology, which does the sample extraction really, really quickly and deposits the reagents on the chip. You sit it in the device, it's affordable, you can carry it in the palm of your hand, it connects to the mobile phone, and it also gives you real-time surveillance. So I started off with this vision, handheld diagnostic, and I hope I've convinced you that it now is possible and we can have multiple cartridges, identifying a variety of bacterial and viral infections that you can plug into a cartridge. And it's a diagnostic you can take anywhere. And I also show this photo. This is an outreach we do at Imperial, the Imperial Festival, of a very young child using technology. I mean, I say this out loud. You can create a diagnostic, but you need to make the diagnostic that it's going to be easy to use and people are going to want to use. And the hope is that if we make something simple enough that even my son can use, then it's going to be rapid and game-changing throughout the world. To all the researchers, I mean, there is no easy way to achieve things. And quoting my good friend Albert Einstein, you never fail until you stop trying, okay? And we can really impact chronic disease. We can really impact healthcare. I mean, a lot of the technologies that we're seeing in our hospitals are still archaic, but as we've created huge advancements in electronic and biomedical engineering. So we owe it to the world to disrupt the norm and create new, dynamic, and innovative medical device technologies. And on that note, I need to say big thanks to my fantastic lab at Imperial, my students and my postdocs, who have been key to this mission in us achieving everything that we're doing. My clinical collaborators in infectious diseases, also my funders and the Census Council for sponsoring my talk. And I'd like to thank you all as well for giving me this audience. Thank you very much, everyone.